pancreas is located under and behind the stomach. When the stomach is removed, you can see the long pancreas with a tapered tail and a head tucked next to the duodenum. This location allows it to easily secrete the digestive enzymes into the duodenum for digestion of all nutrient types. The pancreas has a head which is located near the arch of the duodenum. The pancreas tapers down to the tail, which is located by the spleen. When a slice of the pancreas is removed and stained, it looks fairly uniform with variations of pink. Acini are the clusters of exocrine glands that are stained dark pink. It is from this area that several different types of digestive enzymes are made. All food types can be broken down by pancreatic enzymes. Amylases break down carbohydrates, lipases break down triglycerides, and proteases and peptidases break down proteins. The light pink or pale regions are called islet of Langerhans. These are the endocrine regions that secrete insulin and glucagon. When the digestive enzymes are made, the enzymes are delivered to the duodenum through the pancreatic duct. The liver has a multitude of functions. It is our chemical cauldron and mass filtering system. It creates and processes so many substances that we will only briefly review its main functions. It is important for the metabolism of glucose, triglycerides, and amino acids. It can make cholesterol from the cholesterol we eat, or more significantly, from the excess carbohydrates that we eat. It is the place where most of our plasma proteins are formed, including clotting proteins. Ketone bodies are made here when we don't eat enough carbohydrates. The ketone bodies are a byproduct of the formation of fat from glucose by our liver to keep the brain functioning. Bile is created as a means of emulsifying fats when it gets delivered to the duodenum. The bile is also a great way for the liver to get rid of the waste that it picks up from other processes of removing substances from our blood. Bile contains a lot of waste that is intended to be removed from the body as feces. The liver can also act as a storage depot for glucose in the form of glycogen for easy access when we get hungry between meals. This keeps our blood glucose levels steady. It also stores fat-soluble vitamins such as A, D, E, K, and vitamin B12. The liver is the largest internal organ, with 2 major lobes seen in the image and 2 minor lobes tucked underneath. There is a wide, thin ligament separating the 2 major lobes called the falciform ligament. The falciform ligament tapers to ligamentum teres, or the round ligament, which connects directly to the anterior wall of the abdominal cavity just under the belly button. The ligamentum teres is a remnant of the fetal vessel, the ductus venosus, that brought blood from the umbilical vein to the body and to the liver. After birth, blood no longer flowed through this vessel, and it shriveled up and became the ligamentum teres. There are 2 vessels that bring blood to the liver: the hepatic artery from the celiac trunk, bringing freshly oxygenated blood to the liver. The hepatic artery supplies the liver with oxygen as well as bringing in any body-generated waste for elimination. The hepatic portal vein brings blood from the spleen, stomach, and intestines to the liver. This contains any absorbed nutrients or waste from these regions for filtering by the liver. Once the blood from the hepatic artery and hepatic portal vein have been filtered by the liver cells, the clean blood leaves the liver via the hepatic vein, going back to the general circulation via the inferior vena cava. The histology of the liver shows a repeated pattern of hexagon-like shaped lobules. These lobules contain sinusoids, the filtering cells of the liver. Surrounding each lobule at each corner is the portal triad. The triad contains 2 blood vessels for delivery of waste-containing blood and 1 bile duct to remove the waste. The vessels are the hepatic artery and the hepatic portal vein. Blood flows from these vessels to the center of the lobule. As the blood flows by the sinusoidal cells, wastes are transferred to a channel, bringing the waste away towards the bile duct. This countercurrent is an effective way for diffusion of waste and other unwanted solutes to be removed and enter the fluid that will go out via the bile. Finally, the central vein is where the filtered blood collects. The central vein throughout the entire liver converge until it reaches the hepatic vein to return the blood, now clean, to the circulation. After the sinusoids form bile from the collected waste, it is removed from the liver via the common hepatic duct. This takes bile to the cystic duct for storage within the gallbladder. When a fatty meal is consumed, the gallbladder sends bile out the cystic duct and down the common bile duct to enter the duodenum. The role of bile in digestion is the emulsification or breakup of fats. Like oil on water, fat does not dissolve well unless it is emulsified. Like oil and water again, add a little dish soap, mix, and you'll see the large oil pool become hundreds of small oil droplets. That is emulsification. Bile does not break oil down, it only breaks it up into smaller pieces so that the digesting enzymes have more surface area to chemically digest the fat. Other components of bile are primarily waste products such as cholesterol, phospholipids, bilirubin, and many other substances. The liver detoxifies blood by the removal of circulating hormones and drugs. One mechanism of detoxification is the cytochrome P450 enzymatic pathway. The agents are made water-soluble so that they can be carried out of the body by the kidneys with urine or from the liver as bile. This is an important detoxification pathway that may be affected by certain pharmaceutical drugs. The liver also plays a role in metabolism by storing and releasing glucose. When our blood glucose levels are high, the liver then puts the excess glucose into storage as glycogen. Then, as the blood glucose levels drop, the glycogen is broken down to glucose again. When we have no glucose available for our cells, then the liver can make glucose from fat. However, this process creates ketone bodies as an unhealthy byproduct. Triglycerides can also be formed directly from glucose. This means that people with high triglycerides, as determined by a blood test, may be contributed to that by excessive carbohydrate intake as well as fat intake. The liver also uses amino acids to form a number of plasma proteins, including albumin, proteins used for clotting, and globulins. In desperate situations, the liver can also convert amino acids to fat or glucose to use as energy.